Hello everyone, I'm Sean McVeigh, National Training Coordinator for the Soil Science Division located in Lincoln, Nebraska. I want to welcome you to today's two-part webinar on Soil Health Nutrient Tool, the science behind this tool by Dr. Rick Haney, and the Soil Health Nutrient Tool Project by David Lamb. Before we get further into introductions, I just want to go over our webinar. And yeah, if my attendees and participants, the presenters, don't mess with those window sizes, please. The meeting today is being recorded, and we'll take questions by written question at the bottom of your Adobe Connect window, the Q&A pod there at the bottom. You can type your question in, and we'll take those as they come in. It seems like an appropriate time to interrupt. We'll do that for you. If you want to make the presentation larger, at the top of your Adobe Connect window, there's a ribbon there that has an icon, 4 arrows pointing to the corners. You can click on that, it'll enlarge it and go from there. And in the same way, if you want to minimize it, you can click on that same icon. Make it a little easier to see those screens. That's about all I have for the Adobe Connect issues. The webinar, like I said, is being recorded. We'll have that posted to our National Soil Survey Center YouTube channel. You can search for that. It'll be publicly available within a week. So with that, I'm going to turn it over to Mike Kuchera for introductions.
Okay, thanks, Sean. First of all, I want to thank Sean for doing all the technical support here, and then I'm going to do a short introduction of our two speakers. A little bit of background here. One of the reasons that we wanted to have this webinar is both from the field and from the center perspective on especially how the soils division can support soil health, looking at maybe broadening what we do here at both the center, the lab, we've got research scientists here in the room. So we're interested, I guess, as David and Rick go through their presentations, how we can help support that and provide that niche. Of course, here at the lab, we provide support for the soil survey program, dynamic soil properties, and various other things, and in looking at maybe broadening a little bit into the soil health arena as far as the support services. So that'd be just a little bit of a background. Introduce our speakers. I'll start with David. I've known David for several years now, and David is the director for the soil health division there in Greensboro. And David, his background was he was a DC in Indiana. From Indiana, David moved to assistant for programs, keeping that technical information in programs, and then from there he moved to the center and is currently the director there for the team in Greensboro. The next speaker is Dr. Rick Haney, and Rick has went to Southwestern Oklahoma State, biology chemistry degree, soil science degree from Texas A&M. And his master's degree in soil science, and his PhD is in soil fertility, chemistry, microbiological ecology at Texas A&M as well. And Rick has been with ARS in Temple since 2002 and had various graduate, postdoctoral teaching assignments down at Texas A&M. And one of the things I picked out of his bio here is he's got some background working on a farm before and after he graduated with his MS, and various other things like mechanics, furniture, upholstery, those types of things. So Rick is definitely a hands-on type person, and Rick has been a very good partner in working with NRCS on the soil health nutrient tool project, which is David will be talking about. That project is a CIG funded project, and most of the states in some of the territories are involved with that and doing on the ground evaluations of using the soil health nutrient tool and the recommendations for nutrients to try to get those, better information, better science, if you will, on how that tool can work across the country in different regions. And then the project that David will talk about supports that, and Rick is going to cover the science, which is very interesting to our group here, especially with the research and the lab functions. So with that, I'm going to go ahead and turn it over to David, to Rick first, and I guess you're on.
Okay, thanks, Mike. This first slide is an overhead shot of our research station. We've got a couple hundred acres there, and you can see that the strips are, we've got a no-till, conventional till, side-by-side system with only cover crops as a fertility input. So we're sampling these and doing crop rotation and cover crops to try to get a handle on what we're looking at. How quickly do these systems change? How quickly can we pick up the change in the soil? That kind of stuff. So that's kind of where we started that 3 years ago, I think. But today I wanted to talk about soil testing. Traditionally, we've treated soil as a chemical system, and rightly so. Soil chemistry is much easier to measure than soil biology, so it's no wonder that we went to soil chemistry and the soil physical properties. They're easier to get a handle on. Soil biology is dynamic and difficult, so you can kind of see why we went that way. But the other part of that puzzle is that we haven't had the technology in the lab and the innovation that we've needed to really pursue soil biology. So we need to move forward on that, and we really have the technology and the innovation that we really need to move this deal forward. How we've tested this in the past is just, and all we've really focused on is soil NP and K, because those are the biggest expense on the farm as far as fertility and fertilizers go. So we wanted to really try to be as efficient with that as we could. But we've treated the system, the soil, as a non-living, non-integrated system, and that's just the exact opposite of what it really is. And we focused on physical and chemical, ignored the biological. And then the other piece of that is that we've extracted soil with chemistry that the soil never sees. So we take a sample in the field, we bring it to the lab, and we expose it to chemistry that just, it doesn't naturally happen. And so the other part was we were trying to mimic nature as best we could in the lab. That's really what has been driving this. And the other thing we do is we measure the house and not the food, and I'll go into that. So, but lab chemistry, this on the left side, you see sulfuric acid, hydrochloric acid, nitric, all these acids and different compounds that we've used in the lab since forever, since the 50s and 60s. You get the soil in the lab, we expose it to these different chemicals and try to make sense out of the results. The new way that we do it is that we use water and an extractant that I developed that's based on naturally occurring organic acids. Those are the only two things we use in the lab. So we don't use any of the stuff on the left. Not that we get a total picture with what we're doing, but we get a more isolated picture of what the biology is trying to do. So to start out with, this is just a slide to show the nitrogen cycle, and it's a vastly complex, and you guys all know this, it's very complex, it's very dynamic, and it changes a lot. And trying to get a handle on what it means and how we evaluate it has been a daunting task. However, I think we've come up with some simple things that we can do that will help us get down the road a little bit. And so currently, for nitrogen, most labs test for nitrate nitrogen, that's it. And that's been around. The paper which first started using 2 molar KCl was in 1965. So that was quite a while back. And the other thing I've run across that I didn't really understand is that there's an amazing amount of soil testing labs out there that don't measure nitrogen in any form for various reasons. And that was kind of shocking to me. What we're doing is we're measuring ammonium nitrate, water extractable total nitrogen. We use microbial respiration. We look at the organic N pool, the water extractable organic C to N ratio, a calculation called MAC-WEON. We measure an N-min nitrogen mineralization, and again, we use water for all that. That's all water-based. So that's a very different approach. The same goes for phosphate. Again, we have a dynamic cycle that, to me, phosphate or phosphorus is the Pandora's box of soil nutrients. It's just so difficult to understand. It's difficult to reason out what's available and what isn't. So once again, a very complex system. Currently, you can use a couple of different instruments, an ICP or a colorimetric machine that looks at orthophosphate, and there's 7 different extractants used worldwide that are fairly popular. So you get to choose from those. And we get a lot, when we started in this years ago, we got a lot of questions because we had farmers and ranchers sending labs different, the same sample to different labs, and they were getting very different results. And they didn't understand that and thought the soil testing labs just didn't know what they were doing. Well, I've always asked them, well, what extractant did they use? Well, they never know. And they got to understand that, the amount of phosphate you get out is very dependent on the extractant you use, and especially the strength of the acid or alkali that you use. And so that's why you get these very, very different results. So in our deal, we use both machines. We use an ICP and a colorimetric machine. We use the extractant I developed that mimics the plant root exudates. And again, we use respiration, the C/N ratio. We have a P mineralization equation. We look at the percent of P we get from a water extract versus the percent that's extracted with my extractant. We look at that ratio. We look at the amount of phosphate that comes off the ICP and divide that by the iron and aluminum to get an idea of the P saturation on iron and aluminum sites. We also look at calcium and divide that by iron and aluminum for a ratio to kind of give us an idea of where the pH is and what's happening with the chemistry. Soil health is, of course, you've all heard that term, and it means different things to different people. I've seen a lot of stuff on the internet about different soil health methods. One of the more popular is a permanganate, which is also called active carbon. I think it's a fine test, but it actually lines up better with organic matter than what we're measuring. And it's really not what the microbes see. What they see is the water-soluble carbon, which is what we're using. And organic matter is another popular measurement, which I think is fine, and I think it's great. But that's really measuring the house and not the food. And there's another one for nitrogen mineralization, which is an anaerobic nitrogen mineralization test that's done at 40 degrees Celsius under anaerobic conditions, and it's really not what happens in the field. And you can't tell when you got in nitrogen immobilization from that test. I like the test, but it takes 7 days to do it, and there's some things that it misses maybe. And so our approach has been different. We use the respiration. We look at the water-soluble organic carbon. So we take the soil, extract it with water, and then run organic carbon and total nitrogen on it. And what we found is that's the food. That's the microbes are really chewing on. So we look at that. We look at water-soluble organic in. We take the total nitrogen from the water extract, subtract off the inorganic nitrogen we find in the same sample, and then calculate the difference, which is the organic. We look at the C/N ratio, and then we have a soil health score and a cover crop suggestion. So our approach is a little different. Now, the soil health methods that we've come up with. I said you can tell I'm an Okie and now a Texan, but anyway, we look at water extractable organic carbon, organic nitrogen, C/N ratio, 1 gauge CO2 microbial activity test. We still look at inorganic N and P and K. We look at H3A extractable, aluminum, iron, and phosphate. So these are the measurements that we take, and then we integrate most of that data together to try to understand what we're looking at. So the question to me is soil extraction. We use H3A in water, and why? Well, because that's what plant roots really see. We use water because when I talk to farmers, one of the things I talk about is that it rains water. What the soil sees out in the field is water. It doesn't see all this complex chemistry we've been using and I've been using in the lab for a number of years. So what they actually see is water and a complex mixture of plant root exudates and enzymes and nutrients. And there's this elegance that flows underneath the ground through the root systems, and all the complexity and the microbial and the biological and the dynamic systems that are all playing a part when soils are warm and when they're really functioning. And we need to try to capture that somehow. Now, in the last 50 or 60 years, we've been extracting soil with highly disruptive acidic or alkali solutions, calling it plant available, and it really isn't. And there's been a lot of calibration work done on different extractants and all that, but most of that was done in the 60s and 70s. And so hopefully with the new data and new understanding, we can build on what's happened in the past and maybe do it a little better. And to give you an example of the phosphate, we did an experiment. I haven't published this yet, but these are some of the graphs. And on these are extracts, and that's water on the y-axis in part per million, done on an ICP. And on the x-axis is iron aluminum oxide strips that are supposed to be kind of the gold standard for looking at phosphate availability in soil. And so that's what we compared. I think there's 200 samples in this database. There's probably 70 from the NAPT soil system, and the rest are from farmers' fields around the country. And you can see that water has a pretty good relationship with iron aluminum oxide strips, which is encouraging because water's a pretty neat extractant. However, if you look at the axis, we're talking about 0 to 100 part per million. So just bear that in mind. This is a decent relationship, but water, the bulk of it's below 20 part per million. So this is the same data. This is Malic 3, which is a really popular extractant around the country. But I want you to notice that on the last slide, the water, the y-axis went from 0 to 100 part per million. The same soil using Malic 3 goes from 0 to 800 part per million. So that's a radical change. It's a very similar R squared. The slope is another thing to notice, in that the slope of this is 0.17, and in Malic it's 1.5. So Malic gets almost 10 times the amount of phosphate out that water does, based on the same set of circumstances. So we have to be careful in our interpretation about what we're doing and what we're seeing and what we're actually looking at. This is the same data, which shocked me when I did this experiment initially, using the extractant I developed. I mean, there's a really strong relationship. It's got a great R squared, and the slope is almost 1, which is kind of what we were after, was if we could at least mimic the iron aluminum oxide strips. Because the way you do that test, and many of you may be familiar with it, but you take a gram of soil and you put it in 20 mils of water in a centrifuge tube with that little strip in there, and you shake it for 16 hours, and then you pull the strip out and knock the phosphate off of it and run it on ICP. And so that test is supposed to be pretty close to what natural conditions say, as close as we can get anyway. And so we were really encouraged by this kind of data. And this graph is the same data, but I went back and I've done 3 different derivatives of the original extractant, and I was really clever with the names. I call the original one SE1, which is soil extractant 1, and then I made soil extractant 2, and then now H3A is the final iteration of that research. And you can see that the slope started out 1.42, then I got 1.05. I was getting there, and then the final one is the slope is 0.95. So it's been an evolution of trying to make it better and make it reflect what we hope to see in natural systems. But again, this is the tip of the iceberg, and I fully expect somebody to come up with something better in the next 5 or 10 years. So, but I think this is a step in the right direction, using these organic acids that plant roots actively dump into soil, and try to mimic the natural system as best we can in the lab, because there's always going to be limitations. So, organic matter, we hear that a lot. To me, that's the house the microbes live in, and the water extractable organic carbon is the food they eat. And so what we're measuring is this water extractable carbon, which I've got the arrow pointing to the food. And so within that, and the other nice thing is we show water extractable organic carbon, water extractable organic nitrogen, you know, and there's phosphate on there. And what we tend to overlook, or I've overlooked in the past, is that these aren't separate pools in the soil. They're all combined together. So when we look at the C to N ratios, I think it's a fairly realistic look at what's happening, because if the carbon's there, usually the phosphate and nitrogen are probably there too in the proper proportions. And so my wife made this slide for me because she knows that, well, her opinion is that I'm not as bright as she is, and I can't really argue that. But so she made some pictures for me, so so she said I would understand this better. But when we measure organic matter, I mean, you can use loss on ignition, or you can use this machine that uses combustion. And so these are the very similar machines, but we extract the soil with water for this one, and this measures part per million. This one measures percent organic carbon. This measures a much smaller pool than this one does, because we've had a lot of folks say, well, why can't we just measure organic matter and calculate the water extractable organic carbon relationship? And we can't find one. The pools are too big in organic matter or soil organic carbon compared to water extractable. So we really struggle to find a relationship that you could count on. Now, so to show you some of that, here's a couple of graphs. On the left is soil organic carbon versus soil organic nitrogen. And it's a nice relationship. The slope's about 10.8 to 1. So that's close to that'd be pretty close to what the C to N ratio is. But the x-axis goes from 0 to 5,000 for nitrogen and 0 to 50,000 for carbon. That didn't turn out right. I'm sorry about that. On the right-hand side of the graph, that's water extractable organic nitrogen on the x-axis, and the y-axis should be water extractable organic carbon. Now, these are the same soils, the same everything, but the slopes are very close, 10.4 versus 10.8. So this is a subset of the larger pool, but we're looking at 0 to 80 part per million on nitrogen and 0 to 1,200 on carbon, as opposed to 0 to 50,000 and 0 to 5,000. So it's a subset of the organic matter pool or organic carbon pool, but it's the ones that the microbes, I believe, actively act upon. Is my okie still coming out? So on average, based on that data, which is, that's, I think, 50 or 60 samples in that, but around the different parts of the country, but only 4% of the organic carbon is in the water extract, and 2.9% of the soil organic carbon is in the water extract. So you've got this organic matter, you've got the water extractable stuff, and of that carbon, only 8% is being mineralized by the soil microbes in 24 hours, which is the test we do, which is 0.34% of the soil organic carbon. The point of that is that you can measure soil organic matter or soil organic carbon, but what's actively in play by the microbes is a tiny, tiny portion of that, and we're trying to blow that up a little bit to get a better handle on it.
Rick, do you have time for a real quick question?
You betcha.
Yeah, one of the questions was, we've got a soil that, is mostly humus or stable organic matter compared to one that's got more fresh organic matter, basically, or maybe even a soil that's been abused compared to one that hasn't. What you're trying to do is get a snapshot of availability based on those different pools, right? Is that right?
That is right. And we've seen, especially from soils that we get up north, North Dakota versus Texas, very different. Bigger pools in North Dakota because their carbon's not in play year-round like it is down here, and that affects things, because the measurements that we're making are not designed to be apples to apples across the country. I mean, some of it is, but I think what we're going to find in the future is that by regions, north, south, east, and west, you're going to see differences that are going to apply to those areas, and I'll cover a little bit of that in a minute. This is that same data we were just looking at. that's the microbial activity from the drying and re-wetting and 1-day CO2 on the x-axis. The y-axis is soil organic carbon. You can see there's a relationship there. It's not a very, it's not a great relationship, but there is a relationship there. Now this is the same data, 1-day CO2 versus soil organic nitrogen. Once again, a relationship, not a great relationship. When we take the same soil and look at the water extractable organic carbon and the microbial activity, now the relationship comes into focus. And so we see, that's what led us to believe that this water extractable organic carbon and organic nitrogen is the food the microbes see. And that's really important, I think. These two pools, here's the nitrogen, are really important indicators of where soil is and where, what its potential is and what you can do with it. So there's microbial activity versus water extractable organic nitrogen. I should have thrown a slide up there to show versus total water extractable nitrogen before we subtract out the inorganic, because this relationship gets real fuzzy, and as soon as you subtract the inorganic out, then this relationship shows up. So it pops in there real nice. One of the things that, there's a big push on for cover crops in this country, and I hope it keeps going, because I think it's really a key to the future of U.S. agriculture, because we actually can grow microbial and organic chemical diversity with plant roots. We can. That's where the microbes are. They're around the plant roots. The system's dynamic. It's functioning. It's capturing light, sunlight. It's providing energy to the soil. I mean, there's just a lot of benefits beyond just high yields, our pursuit of high yields. Keeping the soil regenerated and moving, and I think is just a great thing that we could and should be doing. So to put simply, I think if you want to fix your dirt, you use plants to do it. They're dynamic, and they're part of the solution. That's why I really like the cover crops. I want to give you a little bit of history, where we were and where we've gone. We, back in the 1890s, they started to see some of this. Organic compounds were important. 1920, they started to see, CO2 microbial respiration and ammonium and nitrate formation were parallel processes and biological in nature, because there was some question back then as to whether it was biological. And then they started looking at drying and re-wetting soils. They started seeing CO2 evolution, or microbial respiration was a great estimate of microbial activity. And in 1935, I put this one on here because, CO2 evolution was the first day after re-wetting a dried soil. Well, that's what we're doing. And, that was, 80 years ago. They knew about this. So they were starting to understand this. And as things moved on, they got better and better at it. And then Birch did some great drying and re-wetting stuff back in the 50s and early 60s. And then in 1970, from there, we kind of changed our focus from microbial respiration to soil microbial biomass. And then, boy, it was okay to do that, but we just kind of went off the rails with respiration. And we're trying to kind of bring that activity back. There's nothing wrong with microbial biomass carbon measurement, but it's not a dynamic measurement. It's a measurement of the mass of microbes you think is in that soil. What we're trying to focus on is what's the activity. So back in graduate school, I tried in 1995 to publish a paper, drying and re-wetting the soil and look at the flush of 1-day CO2 to estimate N mineralization. That paper was rejected. And this is a true story. It was deemed too simplistic by the reviewers in spite of the data that was presented. And at that point, I became emotionally disturbed. I mean, I got my PhD when I was 42. I came off the farm, and I thought, wow, everybody's going to love this. And no, wrong. Things don't move that fast. So here we are, almost 20 years later, and maybe we're starting to see some of that. This is data that I did in 1995. I didn't have the technology in the lab. It was kind of labor intensive to do this, but what I was looking at was real-time CO2 evolution data. We'd take the soil, re-wet it, dried it and re-wet it, and then I tracked each sample for 24 hours in real time with chart paper, if any of you remember chart paper. And so what I was looking for was the peak of that flush. When you re-wet a dried soil, the microbes start working, and then they start pumping out the CO2, and then they'll peak at some point and then start falling down to a basal line. And all I was really looking for was that peak. But what blew me away was that peak occurred on these 9 soils in within 1 to 6 hours. And as compared to on the y-axis, nitrogen mineralization in 30 days from in an incubator. I mean, look at that relationship. I showed that to my major professor, and he thought, that can't. He said nobody will believe that. But I mean, we kept repeating it, and it kept doing the same thing, and it really opened our eyes. This is the same data with carbon mineralization for 30 days. I mean, I don't know how it gets any better than that. So the point of this whole thing is that, we were looking at these several week incubations and long-term stuff, and we were getting the relevant information within, a few hours or at least 24 hours. And, that was just amazing to us. Because when I went to grad school, they said, well, if you have a dried soil, you have to re-wet it, put it in the incubator for 7 days, and then you can start your measurements. Well, the problem with that was we missed the whole game. That was the current information and the way things were thought of at that time. So, maybe we can move forward. Soil nitrogen, we look at total water extractable nitrogen, and then we break it down into inorganic and organic. And those two things have been really helpful. The first change we see in soils that we've been getting from around the country when they switch to no-till or cover crops is a change in the water extractable organic nitrogen. The ratio of those two seems to be a pretty good indicator that you're headed in a different direction. So that's been a very interesting thing to watch. Since 1965, we've been missing half the nitrogen. This is from, I think, 2013. We ran about 6,200 samples through the lab, and we saw 36 pounds on average across all those samples in the organic fraction and 34 pounds in the inorganic fraction. So we were missing the 36 pounds. And that was something we thought was important to look at because that's already in the water extractable organic nitrogen form, which should be readily broken down by microbes over that growing season. And, that's something that we need to look at and maybe account for a little bit better.
Rick,
I like this slide because, okay.
Yeah, real quick question on that previous slide. You had the organic in. Just kind of reiterate that organic in that's what's available in that first 24 hours, or from the re-wetting of the soil. How does that compare to? There's other organic in the system as well.
Right, that's a good question. No, this is the total nitrogen that we find in the water extract minus the inorganic, which shows us the organic nitrogen. We give you, depending on the C to N ratio of your water extract and the microbial activity, we give you all of that as plant available over the growing season, or a portion of that, depending on, again, the microbial activity and the C to N ratio of the water extract, because sometimes we don't give you any of that as a credit for plant available nitrogen. If you can have a lot of nitrogen in the system, you can have that much organic nitrogen in the system, but if you don't have the microbial activity going for you, or the C to N ratio is too high, we don't give you any of that. So that's something we believe that is your mineralizable nitrogen pool over the growing season, is that organic nitrogen from the water extract. That's what we believe so far.
Thanks.
Yep. This is just to kind of visually show that all we ever really have looked for in the past, myself included, is nitrate nitrogen, and that's it. And we've ignored all these other compounds. And so, there's folks out there that don't like my extractant, and that's fine. But the water extractable stuff, the microbial activity, the organic carbon and organic nitrogen, I think are just absolutely essential to measure for us to get a handle on the dynamics of different soils and different climates. And this is something that we've been doing. I didn't, for some reason, that kind of got cut off. But when we've had farmers and ranchers send us samples, everybody's wanted to know, well, what's a good score, or what's a good number, or what's a good this, or what's a good that. And so what we started doing in the last 6 months is if they send us a sample from a field, we've asked them if they would go take another sample in their fence row, because usually that soil underneath that fence hasn't been tilled, it hasn't had a cover crop, it hasn't had fertilizer applied, it hasn't had anything done to it. And so that should be kind of a little bit of a benchmark for where they are and what they could be doing. So the red bars, for example, these are different states. I think Iowa is the far right one. I've got the slides cut off, so I can't tell, but the green one's Texas, I know. I put that one in there because it's the biggest, because that's I would get fired if I didn't put that on there. So, and I think the red one's Oklahoma, but the shorter one, this is water extracted organic carbon. So the first one is the field, and the second one's the fence. The first one is the green is the field, and then the fence. And so you can see, and the last one I know is Iowa, and you can see that they're pretty close to what their fence row says. So this is a real simple thing to do that I think farmers and ranchers like, because it gives them a benchmark with which to understand or compare what their numbers mean. This is the same data with the soil health score, and you can see, the soil health score is usually higher, quite a bit higher in the fence row than in the field. So, all these indicators point to that. That's the water extractable organic nitrogen. Again, the Iowa sample, which is the far right aqua colored one, is pretty darn close. Here's microbial activity. We see big jumps in that sometimes. But I just think if you guys are working with any farmers and ranchers out there, and if they're running whatever test they're running, have them take a field versus fence. That's a real simple, easy way to do a comparison. All right,
Rick,
soil drying
and didn't update with you on that one.
Run that again.
Our screen didn't update with you on that one.
What are you looking at now?
Like I see it's okay on. Okay, it's okay.
Okay. So soil drying and go ahead.
Just a quick reminder to the audience: if you have some specific questions as you're going through, don't forget you can go ahead and send those to me online. Thanks.
Okay, so soil drying and re-wetting. My focus has been to try to mimic natural systems in the lab. That's a daunting task. That's a difficult thing, but we're trying our best, and this is where we think we need to head. And one of the things that the farmers really seem to respond to is when I point out that plants turn greener after it rains due to the release of N and P. It's not just the water, and farmers get that, because if you want to green up a wheat crop, you top dress it and watch the color change. And so when soils dry out and then it rains, that is drying and re-wetting, and we get this flush of CO2, we get a release of N and P, and that's something we're trying to capture, because I believe that the majority of nutrient cycling is done in this drying and re-wetting effect, and we sure learned that some from the drought here in Texas and other places around the country in the last few years. And our laboratory analysis really doesn't account for this right now. So that's something we're trying to really capture. Again, my wife made me another graph. There's the rain cloud. It rains. It kicks the soil microbes in. They start working on that organic carbon, nitrogen, phosphate pool. We get out mineralized C, N, and P. Plants turn greener. So, there's something here and something we need to try to capture. This is to kind of show, when it rains, you get plant roots get more active, you get these acids. Those are three of the three acids that's in my extractant at about roughly 1,000 molar concentrations: malic, oxalic, and citric. And so it rains. What we do with that is we start measuring these things, and we integrate these things. So we measure phosphate, potassium, calcium, iron, aluminum based on those acids. We look at the percent phosphate divided by iron, aluminum, calcium that I went over. We look at water extractable nitrogen, water extractable organic carbon, ammonium and nitrate, from which we can calculate water extractable organic nitrogen. And all those things on the right go into the soil health calculation, which I'll show in a minute. So, but that's kind of a visual representation of what we're trying to do. This is to show you that this is what it looks like. This is after we shake it for 10 minutes on the upper left. We use both my extractant and water for every sample, and then we filter it, or well, we shake it, we centrifuge it, and then we filter it, and that's what it looks like in the bottom right when we're finished. We take the 40 grams of the soil, put it in 20 mils of water. There's holes in the bottom of that cup with a filter paper in the bottom of it, and it'll re-wet itself to roughly fill capacity. You put the paddle, CO2 paddle in there and get your respiration. And with the samples, we run water on both the colorimetric machine for ammonium, phosphate, and nitrate. The same sample, the same standards are run on the carbon analyzer. So the water sample gets carbon and total nitrogen, and then inorganic nitrogen. The H3A extract gets run on the colorimetric also, and this is a microwave plasma or similar to an ICP. So there's some built-in checks here. Because my water extractable nitrogen better be higher than the sum of ammonium and nitrate, and the orthophosphate better be less than the phosphate we get out of this machine. So we have these cross checks that's built into the system to kind of make sure that we're on the right track, the samples are in the right order, and the numbers make sense. And then here's the respiration. These paddles turn color. That's a real neat system. There's another system we're working on to try to do some of this, but this is a neat system. The paddles react to the CO2. You can put it in a digital reader and off you go. Real simple. This is what the raw data looks like. We have 4 Excel sheets that we end up using, which I won't go into all that, but this is what the raw data looks like. These are the numbers straight off the machines. And then this is a calculation sheet. So there's a series of macros in here to run all these calculations, and all this stuff's open source. We'll send this to anybody who's interested. And most of the commercial labs that are running this test have all this information, which is easily manipulated. I mean, you can change it if you don't like something or don't believe something's right or wrong. So we hit a run button for the macro, and it does all these calculations, and so you can go in and look at what exactly it calculated for this or that. And here's some of those calculations. Again, easy to change. More calculations. And then here's the output file that we send to farmers and ranchers. You'd scroll to the right to see this. Here's your pounds in, P2O5 and K2O that we find. We multiply those numbers times current fertilizer prices to give you a nutrient value per acre, which is kind of fun. Farmers kind of like that. And we have a little run calculator for your fertilizer needs. And that's easily changed. For example, we've got corn set at 0.95 bushel per pound of nitrogen per bushel of corn. If you don't like that, if you think it's 1.2 or if you think it's 0.75, you can change it right there just real quick. So it's very flexible. This is some of the soil health stuff. There's your microbial activity, organic carbon, the seed-in ratio, and the soil health calculation. That number is based off these things. So all 4 of these go into that, creating that number. So this is activity, food, and the balance of the food is kind of how we've set it up. We give you here's the total nitrogen, here's how much was inorganic, and here's how much was organic. That's kind of a neat, that's a neat balance of things to look at. My wife made a tab on here that you can look at that same data graphically if you want to. It's just got a graphic tab, which is really nice, and farmers seem to like that a lot. This is some data that we've done over the years, and I wanted to show you this because we get a lot of questions about the depth of the sample. We recommend a 6-inch sample because most all commercial and university labs use 6-inch samples. All the calculations are based off 6-inch samples, but I wanted to show you here at the very top, that's the same sample. That 0 to 2 inch and 0 to 6 inch is the same sample. And when we start getting too deep, we start diluting the effect of what's going on. You can see the microbial activity is way higher in the 0 to 2 inch than the 0 to 6 inch. So it does matter what depth you do. I personally would like to see us do a 0 to 3 inch sample for soil health purposes, because that's really where the ball game is. I think that's where most of the P and K are, especially in a no-till system. And, I doubt that we'll ever get that pulled off. But these samples below it, 2 tons, 4 tons, 6 tons, are all from Riesel, Texas, and where they've been putting poultry litter on for, I think, 10 years at different rates. There's the rates. But the point here is that folks say, well, can you make your soil healthier? Can you change the soil health calculation? Well, absolutely you can. I mean, it went from 10.57 to 18.19, as you increased the things. Can we pick that up? Yeah, we can pick that up. The carbon picks it up. Microbial activity picks it up. So these things can be done. That's a North Dakota producer who's got one of the higher soil health scores I've ever seen. I won't mention any names, and then the Temple 07 versus 2013 is here, same site on one of our fields that we've been monitoring over the last several years. And our soil health score has gone from 587 to 799 because we've just been using cover crops. One of the plant material centers around the country, again, won't mention any names, but has the record for the lowest soil health calculation so far. And so there's a pretty big range. We've seen some soil health scores over 30. Those are usually pastures that, native pastures. We've seen some of that. So there's a pretty big range in what we can see here. These are the guys that I work with in the lab. That's Buddy on the left and Chris on the right. They do the work. I'm just the brains of the outfit. That usually gets a fairly large laugh whenever I say that in front of these two. So that kind of tells you where I'm at. This is them hard at work. We have a set of monitors that we can watch all the machines from, and we're trying to drag our lab into the 21st century, which hasn't been easy. That's a look at them. Those are the machines running. Those guys are so involved with data entry and try to analyze all the data we're getting and talk to the farmers and ranchers that we had to set it up so we can watch the machines from in there. So we had places where we'd have a machine quit running and we wouldn't know it for a couple hours because we've been in there doing that. So we tried to set it up better. This is something that I'm working on that I built for research purposes, where we can monitor CO2 respiration for soil microbes in real time on 42 samples instead of one at a time. So that's something I'm working on, and hopefully we'll have that up and running really well this year. This is kind of shows you what that looks like. There's 4 samples. We dried and re-wet it, and we ran. You can see that the green one, it took about 10 hours, 12, 13 hours, and it peaked, and then it kind of held it. The black one peaked within the first 5 hours and then came down. I mean, we're getting all kinds of different readings here. We got what we call slow risers and fast risers, and I don't know why yet. It's something else I want to look into and hopefully collaborate with some folks on looking into, because I think this is really important. Here's a look at some, I think there's 12 samples in here. And you look, I mean, boy, why did those top 2 flush out pretty big in the first couple of hours and then drop and then go really big again? I mean, those are very dynamic soils, and something's going on there, as opposed to some of these on the bottom that flush out and kind of just maintain. Some rise slowly. There's a lot of stuff here that I'm really interested in and think is going to be important in the future for some of the research we might do. This is a look. These are slides that are showing water extractable inorganic versus organic nitrogen over the years. We ran 3,000 samples in 2012. We ran, I think, 6,000 in 2013 and about 5,800 in 2014 from around the country. And the first year, there was a wide difference, 36 versus about 47 pounds of nitrogen in the inorganic form versus organic. In 2013, they got closer, and in 2014, they flipped. We're going through the data right now, but what we're looking at, the biggest influence on that the reason for that shift has been the influence of cover crops. We've got a lot of producers. Over half the producers we work with are using cover crops, and have been using cover crops. So hopefully we can see these kind of trendy changes in the system. This is a phosphate, inorganic versus organic, which, a phosphate's a different animal, and it's not like nitrogen. I didn't expect to see a big change here, and sure enough, we didn't. This is microbial respiration, or one-day CO2, so that's on the upswing. Of course, this is going to change by year, but, this is the sum of 5,000, almost 6,000 samples in 2014, 6,000 in 2013, and about 3,000 in 2012. But, we want to be able to look at these longer term trends and hopefully make some sense out of it. This is my favorite one I fooled the boys with. I said, look at that huge change in water extractable organic carbon. Looks like a big smile. But if you look at the axis, there's really no difference because it's 212 to 217. So there was no difference at all. And so sometimes, we have to keep things in perspective, which is, why the smile, I guess. Anyway, that's all I have, and I'll answer any questions you might have to the best of my ability.
Okay, hey, thanks, Rick. I've got quite a few questions, actually, so go through several of them here. Some of them are more about the test itself and some of that, some of them about the interpretation of the test.
Okay.
One question that came in related to the actual procedure is the centrifuging done just to ease the filtering process?
Yes.
There you go. Simple answer. Okay.
Well, let me address that a little. When we take those samples and run them on ICP, that's a pretty small tubing, and we really got to get everything out of there because we've had that can be an issue. But yeah, that's really just to clean the sample up.
Okay. Several questions, and we're kind of putting our hat on of our traditional soil tests that we do for NP and K. But
sure.
in the case of where we have real higher pHs, how it affects phosphorus compared to water extract, or real low pHs, how that affects, where we get tie up of phosphorus that way. How does your test, do you make any adjustments for that or not?
Not really. The soils that we have here in the Blackland area that's on this farm are pH 8.3 and 55% clay. And the only thing about my extractant that is really kind of unique is that if you extract any soil with Malic 3, it's going to drive the pH of the extract down to about less than 4. And if you use Olsen, for example, on the other end, it drives everything to 8.5. So it extracts the soil at 8.5. Malic extracts everything below 4. Mine will move with the pH of the soil. So because we were at opposite ends of extremes there, I thought. And so I don't know of any extractant that does a great job of extracting phosphate from high pH soils. Mine underestimates the amount of plant available phosphate we've seen. But, I don't know yet how to address that. Plants obviously do know how to address that. But I don't know why. My guess is the organic acids that plants use through their exudates in high pH soil is different than what's in my extractant. But that's a soil by soil, region by region deal. So yeah.
Okay. Another quick question is, can the nutrient value in dollars in the output, can that be mimic the changes in MP and K? Can we change those values?
Yes. You can change everything.
Once you get the spreadsheet, you can basically assign whatever values you want.
Yes.
Okay.
Well, the other thing we do that I kind of glossed over is we don't give you 100% credit for the nitrate. We're only giving you 70% credit, because I don't know of anybody that believes that 100% of the nitrate you take when you take a soil sample is going to be plant available. So we were conservative, and we give 70% of that number instead of all of it.
A couple of the questions we have are relative to how some changes either in the time of the year affect the recommendations and results. For example, the fall versus the spring, for say here in the Midwest as an example, where we go dormant. And And then also how micronutrients can affect biological activity and how that's accounted for, or any ideas there that should be recommended.
The sampling fall versus spring, I've had many farmers send me 2 or 3 samples a year over the course of the year, and they invariably call and say, my numbers are changing, and my answer is always, thank God. It's a dynamic system. They should be changing. But we've had some guys up, that up north.
Go ahead.
Go ahead.
I was just going to say, if they wanted to compare changes from year to year, they'd want to do it in the same time of the year.
Yes, I think so. And up north, I think probably a fall sampling is better than a spring one, because, you know, like you said, you're going to be pretty much dormant from fall till spring. And, you know, if you shut the system down, those numbers should still be applicable in the spring as it's coming out of dormancy. And then the micronutrient thing, we're not, all these tests aren't designed to do micronutrients. That was never our focus. We just wanted to focus on NP and K. Micronutrients are a whole nother question. I do believe, though, that if you have the organic matter, generally speaking, micronutrients aren't going to be an issue if you've got decent organic matter for your region.
And, you know, the biological activity and the impact that those micronutrients have on it, that's kind of somewhat accounted for in the biological portion of your test. Is that?
Well, it should be. I can't definitively say that, but I tell you some of the things that we've seen. We've run over, well, about 17,000 samples over the last 3 or 4 years, and we are starting to see some trends, but mainly, you know, if you've got low carbon, you're going to have low microbial activity. That's just a no-brainer. That just works. But what we've seen now that we're getting some of the fertilizer stuff back is that the higher concentrations of fertilizer that's put out seems to knock the microbial activity back a little bit in fields that are, you know, fairly side by side, and one put on a lot more than the other. We're starting to get that kind of data and see that, and that's really going to be interesting. I have no data on how micronutrients affect microbial activity, but I'm sure somebody knows a lot more about that than I do.
Just a real I got 2 more questions, and I'll try to address some of the others in the chat box that people are also asking. But one is on, you know, most farmers, most land-grant universities, we do our recommendations from standard tests, especially like phosphorus, potassium. We use indexes and tie that back to the likelihood of crop response. The intent of this tool isn't necessarily to replace those tests. It's going to be another tool in the toolbox, so to speak. Is that an accurate assessment?
Yeah, until we get a handle on what all this means, you know, and there's some funding given to land grants to try to test some of this stuff out, and maybe we can recalibrate the 30 or 40-year-old relationships that we've been using. You know, this is well, Ward Labs, for example, runs this test, and last I heard, if you ran this test, they would give you a traditional test also, and so you could look at those things side by side. You know, so hopefully we can get people to think about soil health and how that's impacting nutrient availability, and what, you know, we're trying to pull back and see a little bit bigger picture than what we've been used to seeing, because we've got to get biology in these commercial and university labs if we're truly going to try to understand what we're doing and be as efficient as possible with the nutrients we're using.
Last question, and then some others I'll try to address on chat box. But cover crop recommendations, you've got 90% legume, 10% something else. Are those intended to be general recommendations? I really don't look at the previous crop history and trying to change functional groups and some of the things we recommend on cover crops.
That's correct. That's a general thing. That's based off of your soil health score. That's also easily modified. If we figure out something, we can. So it picks that up, and it'll say, well, why don't you put a few more legumes in this mix than just grasses? because the drive initially years ago for cover crops was all about grasses. And boy, I like the legumes in a mix because they give you that nitrogen, you know, that costs quite a bit of money right now. So that's just that's a suggestion based on your soil test data that we gave you. And, you know, that's open to interpretation and whatever. We had people calling us saying, what should we plant at what rate? And we're like, no, that's not what that is.
Right, yeah. I would think that the carbon to nitrogen ratio would be important too. You know, if you have more carbon to nitrogen, then the legumes would be recommended probably over the grasses. Is that?
Exactly. Yep.
Yeah. Okay, we've got several other questions coming in. I think we're going to go ahead and get started with David. And then Rick, if you could hang on, there may be some questions when David's done too that we may need to do, and the time is creeping up on us. So.
Yep, that'd be fine.
So go ahead, David.
Okay, and again, Rick, I just want to say I appreciate your presentation again. Every time I hear you, I learn so much more. So, and I can see you're constantly trying to evolve and understand more, and give you credit for that. So. But what I've been asked to do is talk about the Soil Health Nutrient Tool Project, you know, and what it's about, and why we would get involved with a character like Dr. Rick Haney down there. So that hopefully by the time I get done, we'll be able to answer those questions. So, but again, I always like to talk anytime I get a chance. The whole concept of soil health is what NRCS is advocating. It's the idea of soils can have a continued capacity to function. So soil to function as a vital living ecosystem that sustains plants, animals, and humans, and I might add has a minimal amount of impact on the environment. And those functions we need, nutrient cycling is key, and water, whether it's infiltrating and being made available, are the two key functions that most farmers really don't understand and don't have an appreciation for, and it's through projects like this that we're trying to educate them on that. Of course, we want our soils to be able to filter and buffer out those pollutants that we might apply to them. We've got to be able to cross the fields without rutting them up. That's a good thing for farming. But the key, what drives all these soil functions is this habitat, you know, and we go out and we talk and we try and get farmers quit thinking about themselves as farmers, but as habitat managers, because that's really what they're doing. And trying to get them to understand that the actions they take on a day-to-day basis when they're farming directly affects the type of microbial activity that's in the soil. And they don't realize they're doing it one way or other. They're managing their habitat. What they don't understand is that they can be changed and manage it in a way that improves those soil functions, that we can get nutrients to cycle, water to infiltrate, and those types of things, and have a reduced amount of impact on the environment. So that's kind of the key. So what was happening when Dr. Haney met Mr. Archuleta and got Ray convinced that this was a good thing. Ray was going around, he was telling everybody Dr. Haney was offering this test for free and it would solve all the problems of the world down there. And where we got involved, and so I think his workload went up immensely from that. You know, anytime you say free in some sentences, that tends to generate work there. So, but what we wanted to do, I guess, as an agency, what we were challenged with is, you know, how does nutrient cycling impact our nutrient management standard? If we have soils that Dr. Haney showed that we were missing about half the nitrogen, and as our soils start to function, does that mean if we continue down the same path that we're going to be encouraging over application of nutrients, nitrogen in particular? And I throw this out there. I don't know how many of you have been followed the press up in Iowa. Interesting article came out over the holidays where I believe it's, and you need to Google this to validate what I'm saying here, the city of Des Moines is suing the drainage districts in the Raccoon River, I believe it is, where they get the primary supplier of their water, because their tile water is polluting it. They think that's a point source discharge. You think about that, and here we've been practicing nutrient management for so many years, and we're still having issues. So how come that is? So that's one of the questions we wanted to answer. The other one is, you know, what's the quality criteria for soil health management systems? Can we establish something? Is there some way to actually measure what's going on in the soil, or do we have to rely on models and interpretations like that? The third thing here is, how do we develop and justify a multi-species cover crop mix? I was at a meeting last week in Delaware. They're spending $70 an acre on multi-species cover crops. How many of us out there think that's a bit too much? You know, how can we justify bringing that down? And how do we know this whole idea of grasses to legumes? So if this test can give us some assistance there, that's a good thing. And then is there just a simplified tool that can give us an overall soil health rating? So those are some of the challenges as an agency that we're hoping through this interagency agreement with ARS that we'd be able to help to begin to answer. We won't get all the answers, but hopefully start to do that. This is what our objectives are. We want to improve the nation's soil health by enhancing understanding of these processes, the chemical and the biological soil tests, improve soil function, the impacts of fertilizer efficiency. All these things we're looking at trying to do. Again, the impact of cover crops. You talk to folks. We as an agency have done a tremendous job of measuring the physical and chemical properties of soil. We know an awful lot about plant growth above ground, you know, the biomass production, the ecological site descriptions, all those kind of things. But we virtually have no information related to what's going on in the biological, the soil biological community below the ground. And hopefully we can start to gather some these so we can compare one system versus another and how it impacts what's going on below the ground. And I really love the one slide Rick showed there. Then was like a 4 or 5 year time period. You can just visibly see the difference in his soils that he's been able to do down in Texas, which the last time, remember, it's down south, got high temperatures, a lot of humidity. We're not supposed to be able to make those kind of improvements that quickly. And then the last thing there is to develop a national database to help to build and be able to validate and calibrate some of these measurement tools out there. So that's what we're trying to do. This is some of the deliverables that Rick's going to give us. He's going to give us a national database. We're going to look at some potential savings or maybe efficiencies of usage of fertilizer. Going to get a tech note out of it. He's going to provide training. I don't know how many times he's given this presentation, but again, to continue and educate our folks and the conservation partners and agricultural producers out there, and then answer a bunch of questions out there. What are we doing? Well, the first thing is we're not validating the science behind the test. I think what Rick has been able to demonstrate through his presentation is that he's got the science, he's got all that information. So we're not going to challenge that. What we're trying to do is help gather information to validate and support the soil health assessment, maybe the nutrient recommendations that come out of the tool itself, that we can adjust those, maybe see some regional variations or what have you based on management systems out there. We want to be able to help evaluate changes in soil health as we start to measure the respiration that's taking place under different management systems, under different parts of the country, under different soil health management systems. We want to collect data and be able to look at that. We want to start to look at the economic and environmental benefits that can be achieved. I mean, I just think about it. If we've been let's say we cut back on 30% nitrogen out there. Just think of the potential environmental benefits that could have as we continue to struggle with the hypoxia zone and the challenges that they're having in the Chesapeake Bay. And then we want to provide research and education to farmers and ranchers and through our NRCS field staff and personnel out there. What's our role? Well, we're going to identify a project contact within each state that's participating. And the states are going to look to have 10, 20, 25 farmers or ranchers participating. We're not only looking at the cropland side. This is equally as valuable and exciting on the grazing side, whether it be pasture and or ranch land. So we're going to have them select those based on their criteria. We didn't want everybody to be practicing a soil health management system. We were hoping they get a variety. We were hoping to get people that may be starting and then follow the recommendations, start to improve over the course of the project, a 4 or 5 year project. Of course, we hope it can explain the project and explain the results and those types of things. So that's what NRCS's role is. This is what the farmers agreed to do. He's agreed to provide a sample on a yearly basis. He's going to follow standards testing protocol where you go out and gather 20 or 25 samples and put them in a bucket and mix them up and send Rick a sample there. Trying to do that from the same field, following under their normal rotation, and then we hope obviously we'd like to see them utilize the information that came from the test. So if the fertilizer recommendations that they're provided, we'd like to see them use those and then be able to track the results as it grow around there or go throughout the project. This is what Rick's going to do down there. Yeah.
Hey, David, real quick on that previous slide, are most farmers, are they doing the status quo, if you will, with normal soil testing and doing some comparisons side by side or not?
Yeah, well, I can't say most because we've done a good job of sending the samples in, Mike, but we have not done a good job of getting the data back, which I'm going to harp on here in a second. But I'll show you the data management collection sheet we put together. We did ask for their most recent soil test information, and something that's 1 or 2 or 3 years old. We don't want something back from the 90s in there, but we do like that, and we are encouraging to do that. Now, one of our challenges is we can't, since we don't have a funding mechanism to pay for that, we're kind of at the discretion of the farmers. But most of them are interested in comparing that, and I really found that interesting. What Rick said about the Ward's lab offering samples to compare. I think that's putting it right out there and let folks do their own evaluation. But we are encouraging that, but it's not necessarily a condition of participation.
Okay, thanks.
This is what Rick's going to do down there. And then, again, it's just standard. Everybody wants to know about the soil sampling protocol. And one thing I found is interesting, again, it varies across the country, the size of the sample. You go up in the New England area, and it's good for 5 acres. And you go out there where you're at, Mike, and it's good for 100 acres. So whatever is protocol for your location, we're asking folks to try and do that and to get it to mimic standard soil testing as much as possible. To date, it's been kind of interesting. One of the reasons, Rick is a research lab down there. For them to handle 5, 6, 7,000 samples is admirable. But a couple things, they're not geared up to do that. So one of the things was to help them kind of get a control a little bit on some of the sampling that's coming in. And then the other thing is to gather data. And I'll show you what we're gathering here in a second, but right now there's 40 states participating. This is voluntary, didn't have to, so there were a few states, due to some logistics such as Hawaii. You can't send soil from Hawaii to Texas very easy. But anyway, we got 1,330 samples. You can kind of see the breakdown, about 1,100 for cropland, a couple hundred for pasture. And then as we speak, samples for 2015 cropping season are being sent in for evaluation. So anyway, we developed a data collection sheet, and it's kind of a fine line between asking for too much information and then not asking enough. But what we wanted to do is get enough information to evaluate the nutrient content. We wanted to be able to kind of, again, look at the soil health rating and then the cover crop recommendations. And we actually have a SharePoint site that the NRCS people that are involved can enter the data in. This is kind of what it looks like. You go in and you register your sample. Again, that's where the 1,300 plus samples are registered. And then this gives you just a flavor for some of the data, the 2013 crop, 14, the planting date, the management information as far as related to tillage. We decided to have a stir rating rather than trying to come up with this lengthy list of tools out there. We know that a stir rating of less than 20, you're going to be really limited in the type of tools you use versus 80. Soil test information that's available, the nutrients, the timing, the method, and the material used, whether it's for the N, P, and K, whether you use manure, cover crops, whether it's a grass legume, or some kind of combination. Gathering a little information related to termination and estimated height, so we might be able to estimate biomass, rainfall, irrigation, and then I must have left off there. We obviously want to yield on that as the final bit of information. This is what we're asking for the grazing side of things. Similar information, soil texture, forage group that that would be your pasture or your rangeland would be composed of. years since it's been tilled, and on down the list there. So
real quick is I've got a note here that some people aren't seeing the screen. I guess we're kind of to the point. We'll just continue on, but we're okay. We're okay. There's the other one we lost the screen. So,
well, I think I kind of covered verbally what I was be showing on the screen there, Mike. So, but I guess the key here is I'm looking down your list of participants. I see a few state contacts on this list, and I would like to encourage folks to get the data in. Now's the time to gather. I think I looked at this morning, out of 1,300 and some samples, less than 200 have actually had the data entered. And Rick can't do his number crunching until we get him the data. And I think some of the numbers that are going to come out of this, based on this array of information we're gathering, even for the first year, is going to be interesting. And then, of course, as we build this database over time, more and more patterns and gaps will emerge, and we'll be able to show that. So, again, we're not soliciting new applications. We want to keep the same 1,300 or 1,400 registered sites. So we're not really looking to add another 1,400. In fact, we're not looking to add any more unless it's extenuating circumstances. Folks can contact me. And with that, Mike, I'll end my comments and turn it back over to you.
Okay, thanks. I've got just a couple of questions.
And if you would, go ahead and show us your contact information.
I can do that right there.
Yep. Okay, one of the questions we had is, the goal of this project, and then the science as well, the differences within regions. Are you looking at trying to evaluate if there's what the regional differences are, and specifically like your soil health score or nutrient recommendations that would come out of the test?
Yeah, obviously that's what we're trying to do. I don't know, Rick, would you want to expand on that anymore, or have you got anything you've been able to determine so far with the few samples?
Yeah, they're going to definitely break out by regions, and we'll be able to, and that's why the Excel files were built the way they were and the macros were built the way they were, because regionally we're going to see some differences in some things, and we're going to be able to account for that and adjust for that. And after a couple of years, I think we'll have enough data to actually define those regions.
Okay, similarly, we had a question about irrigated versus non-irrigated. Is there adjustments for that, or no?
Not yet, there isn't. We're just now finding out. We've been receiving samples blind for the last 2 years, and then David and them wanted to start doing this, which is way better, because we haven't known where the samples have come from most of the time, or what the management was. But yeah, right now we have a very, very limited data set on what's irrigated and what isn't, and that's something we can look into. That'd be something interesting in the future.
Okay, I think that's most of the questions that we had. Anything else that David or Rick that you missed that you want to share?
Yeah, our phone systems have been down for 2 months, and it's probably going to be 2 more months before we actually have a phone. So if you want to contact me, use the email. People get a busy signal and they think I'm that busy. Our phones are down.
Yeah, that's I guess we need to up the amount that you get for the interagency agreement, huh, Rick? Get you got a new phone down there. No, okay. The other thing I would say, Mike, is again, Rick's done some fabulous work. It's getting a lot of interest out there, and it's kind of kicked the hornet's nest, which is fun to see. We're starting to look at some things, and starting to look at that biological component that's been overlooked for so many years, and and again we're gathering data, and I think this thing will look a lot different in 5 years than it does right now. But unless you start on that path, that's why we call it research. Is we're trying to figure out something we don't know, and the answers will come as we go along. And again, I want to encourage everybody. Taking that test was good, but unless we get the data back from you, we're only halfway there. So I'm going to continue to hound and send emails and do what I can to get our folks to get get that information in, because that's really the guts of what, that's the other half of the project that's very, very important.
Okay, we do have, I guess we have a few more minutes yet, and there are several questions that came in, so I'll go ahead, and for those that want to stay on, we can go through those. A question came in, said that some people that are working with private labs that utilize this tool, and by the way, I'm aware of about 10 different labs out there. There's probably more that's come online. But they say that if you take a field and start with no cover crop management system and move to a cover crop management system, you should see a general decrease of soil health, nitrogen, carbon over the first couple of years. What do you see there? Is that more of just a tie-up of the nutrients because you're moving to a cover crop or no-till system from where you were? Is that similar to the transitions we see in no-till systems, or is there, you see anything along those lines?
No, that's right, and that's what, you know, that first slide I showed was our 200 acres here, and we saw the same thing. You know, everything kind of backed off a little bit because you're plowing that disconnect cover crop in every year, and you're just injecting a bunch of food for the microbes, and the no-till system is going to lag behind until some of that stuff naturally breaks down and gets into the system. So yeah, that's a common thing that we see when you start that transition. This is our third year, or the start of our fourth year, and they're now about even again. Matter of fact, the no-till's getting a little bit higher on some of the carbon and nitrogen, but it took about 3 years to get it back.
One of the things that I would share, doing quite a few field sessions where we do in-field testing of nitrates, phosphates, for example, right after they do tillage, and next door we have an untilled or cover crop field, the nitrogen and phosphorus levels spike. So you see some of that mineralization happen short term as well.
Yep.
Question here is should we be, with our standard tests, especially for phosphorus, potassium, liming, those types of things, is producers will use grid or zones to do, which are smaller areas, to fine-tune their application of fertilizers. Do you recommend that with the soil health nutrient tool at this point?
I don't, because and I mean, if we're going to do grid sampling and, you know, take a sample every 2 or 3 acres and miss half the nitrogen, I'm not sure what that, where we're going there. I think we've kind of put the cart before the horse in some of this, because we're still just going off nitrate nitrogen, and there's more going on there than we think. So, you know, that's something we'll try to understand in the future and deal with. I personally wouldn't spend the money to have this test done in a grid sampling, because it's expensive, unless it was a real small, you know, 5 acres or something like that, or 10-acre field. But, you know, I think we need to get a handle on what's really going to be available, as opposed to let's measure half the nitrogen and precisely apply what we think it needs.
Okay, this question, hopefully you can answer it. Is our data being uploaded to the Soil Carbon Coalition or with similar partners? Are we providing that, or is there any effort there?
Not yet.
No, not yet. No.
Okay.
We're having trouble getting our data in, let alone giving it out to other
people.
That's
what I figured. How many years? The question is, how many years are we going to gather data, David, with the farmers in the project? Again, you probably covered that. But.
Yeah, we're hoping to do it for 5 years. So again, subject to continuing funding, that type of thing. But that was the goal when the project was originally presented.
So it's going to be done yearly regardless of the crop. For example, if you're in a corn, soybean, wheat rotation, you would do it for each of those crops each year. Exactly. And whether,
Rick could probably comment on that, whether you would need to do it that intensively down the road, I don't know. But for now, that's what we're trying to do, yes.
Okay. Very good. I think we had a really good set of questions. I kind of congratulate the group for everything that you've sent in. Sean, you want to close up?
All right. I just want to thank everybody for your attendance and participation. And the webinar has been recorded. We'll get that posted to our National Soil Survey Center YouTube channel and be publicly available through a